Well, good day, everybody. Thank you all for joining us this afternoon. It's afternoon for those of us located in the Eastern the time zone of Canada. And wherever you are, have a very nice, hope you're having a very nice day. My name is Valerio Hoyos Villegas. I am an assistant professor of plant breeding and genetics at in the Department of Plant Science at McGill University. We're based on the west side of the island of Montreal. We have a lovely day today. And today my student Jennifer and myself are going to host a workshop on plant breeding simulation utilizing a long-standing tool called QGENE. QGENE, more than in my lab, we use QGENE with not very regularly, because as you will see in the coming slides, QGENE is a tool that you utilize with a strategic intent, and so it's in that sense, its purpose is to determine the major generate the information to make major decisions on the direction and overall strategy of a plant breeding program. So we'll talk about tools that you use more regularly as well in upcoming slides and workshops that we actually have planned for you. So I'd like to acknowledge the team that has been involved, my group here at McGill, as well as our group that started it all at the University of Queensland in Brisbane, and they involve Sophie, Vivi, Wenxi, Mingshu, Ian, and Kay at the UQ University of Queensland Brisbane campus in Australia. So I'm very happy to be hosting this workshop through the Education Committee of the National Association of Plant Breeders organization. So let's get started. And I hope you've been paying attention to my little GIF here, which is on the top left corner of my slide, which represents the surface response of the allele fitness landscape, which is sort of a representation of what we intend to do with simulation and population improvement in plant breeding programs. So QGENE is basically a simulation platform for modeling breeding programs. And it depends on stochastic functions to perform standard simulation. Okay, so as opposed to other tools, which we will look into later on in the fall, it is very much based on the random models and the extraction of data or simulation of data from simulated distributions rather than existing distributions, right? So in that sense, it's important, or it's useful to simulate a plant breeding program, because a plant breeding program is a complex system. We can in that same complexity, we can apply models that are not fully adequately addressed or fully developed in quantitative genetic theory, and we can test whether their assumptions hold to some extent in a breeding population. Okay. So I say here that the simplifying assumptions are made in quantitative genetics, and we can render these theories in a mathematically or statistically tractable environment, such as the simulation environment. And we want to find out whether these assumptions are, as I said, are actually met or not in reality. So therefore, it is useful to have a strategic tool as well as a tactical tool to assist in the decision-making of a plant breeding program. We can, with this information, we can then determine the impact of, for example, new tools. So the adoption of new tools, particularly in plant breeding technology and science, depend on, well, primarily to the interest of the breeder, to the relative increase in the rate of genetic gain, and obviously their association with resource utilization, the cost effectiveness. Okay. So therefore, delta G, or rate of genetic gain, can be assessed using a few approaches, but one of them is basically using prediction equations, which is with real-time trial data, so data obtained through historical trials, which is most certainly a tactical tool. But we can also use it, predict it or assess it with a range of prediction equations together with a software-based simulation platform. And I see there's some questions already coming up. If you have any questions, do not hesitate to put them on the chat, and I will do my best to answer them. But let's bear in mind that this is a two-part workshop today. I will be doing this introduction now on the overview of plant breeding simulation and their impact and application, and then my student Jennifer, which I will introduce later, she will be taking over the hands-on part of it. So there'll be plenty of space for questions. So the delta G can then be assessed that way, and so in the benchmarks of plant breeding, we have first and this is a model that is was originally developed by my colleague Sophie here, which you will see why I have that picture there on the bottom left. But what I would like to outline here is that we can consider this environment, or this the experimental approach to developing tools or developing information to make decisions in plant breeding as this environment where we have a plant breeding model, and we can, in this plant breeding model, we can generate a range of genetic gain scenarios, and we can measure these scenarios in either breeding value or percent genetic gain. Okay. I think, do we have a hand raised? No. Then with, say, for example, QGENE, we can then perform simulations, and we can then estimate what is the potential gain that we can obtain based on these different posed scenarios. With a tactical tool such as DeltaGen, which was developed by Zulfi Jahuver and Dongwen Luo in Australia and New Zealand, Ag Research at the University of Queensland, we can then utilize that same information to perform tactical decisions, or utilize existing information to then calculate the expected scenarios, right, based on real information. Or we can merge the two, okay, the simulation capacity and the tactical tool to understand what is the gap that exists between the simulated condition and reality. So this is what I mentioned here. We can just consider all of this as an environment that enables us to obtain information and then either perform decisions or stimulate new questions that may lead us towards closing the gap in achieving an expected, a desired outcome, such as a particular rate of genetic gain. So, in a more sort of broader overview, we can then take those same two components, the breeders' decision support tools, and the simulation capacity by the various tools, in one of them being QGENE, another one being DeltaGen, and they have inputs that would go into this breeding model, which will tell us information about the realized potential genetic gain scenarios in whatever units you wish to look at, the breeding value units and units of whatever trait units that you're wanting to improve, or percent from the attainable percent gain from the overall simulated environment, or the number of individuals across the population and how their mean performance. And so we can post it in the percent relative to the overall population. And this information then, for example, we can tie it in with an economic model. And then this information goes back to that breeding model and then gives us information about the resource use or the cost to attain that particular rate of genetic gain. At the same time, and I put it more in the distance because there's less connectivity between these two sort of environments, we can then utilize other existing models, for example, that operate more on the phenotypic response or the crop response in the biophysical context in the case of management or environmental scenarios. And we can then link those in with the plant breeding model to ultimately have a much broader overview of not just the genetic component space and the genotype and the phenotypic response effect, but also the broader response in terms of climate and management decisions, and then tie all that information in as well with the economic model. So just some examples of where QGENE has seen application. I'm going to enumerate a number of papers here. So this paper, for example, we can application of identified QTL marker associations in rice quality enabled the authors, or there's a study that enabled us to understand what is the desired or the necessary genetic architecture to undertake a particular breeding approach. Okay, so what are the resources, or what is the there's a structure of the population and the required conditions that need to be met in order to perform QTL integration to its and we can, and then that experiment, computational experiment, will allow us to understand what is the feasibility of it. We can use QGENE also to, for example, create or compare new variance components that are not normally observed, for which we say we don't necessarily have data. We can pose these new assumptions to then create these new scenarios and determine whether changes in the population structure or the variance component structure of the breeding trials or whatever environment it is that we're simulating how that might affect or benefit or hinder our progress. We can, for example, also pose additional conditions or components to our genetic architecture and determine whether it is feasible to then whether more complex scenarios or less complex scenarios would enable us to make perhaps more progress. So, and this paper, they wanted to understand whether the discovery of a series of QTL on coleoptile length would also enhance performance in breeding. We can also use it, of course, to strategize the implementation of new tools such as genomic selection. And in this paper, which I'll talk about in further detail, we can, of course, use it to then, with all that information, say from all these this background information that we have gathered, we can then start conducting gap analysis to determine what are the potential actions that can be taken in order to enhance genetic gain. And finally, and most obviously, we can, of course, compare breeding strategies, which is one of the most basic functions of QGENE. So I'm going to go into a case study of perennial ryegrass. This is a paper that the team released a few years ago, and where we essentially posed three different scenarios of genetic models. Additive, dominant, and dominant. And then we took all of the existing QTLs that are reported for growth score or rate of growth and biomass in the case of perennial ryegrass. And then we post all the different combinations of the genetic models, 3 heritabilities and 3 initial parental population sizes to determine what would be the most adequate strategy to follow and between among HAPSIBs and among and within HAPSIB selection. Okay, so these are just some conditions that we'll go further into when the hands-on part of the workshop. But essentially, what this means is we're running enough models and enough runs to allow for sufficient repeatability and convergence of the models being simulated. And then some of the outputs that we can obtain, and then we'll look at this at length, are of course, the fitness or the genotypic value, the change in phenotypic value, which is our simulated delta G from cycle to cycle, as well as the frequency of favorable versus unfavorable alleles across cycles, and of course, a measure, a related measure of this, which is the Hamming distance, and Jenny will talk about this as well. So this is a simple, 2 simple strategies or scenarios that this case study had. So where, for those of you that are not familiar with forest breeding, these are population improved species. They are very much, they operate in a closed or semi-closed recurrent selection program where the end product is a synthetic polycross made up of anywhere between 5 to 30 parents. Okay, and so these are large populations, which essentially the end product is not a line, but rather a population per se. And it's difficult to know what is the as you can surmise, the individual contributions of those potential final parents are hard to establish, and the route at which we end up determining the optimal breeding value or the optimal contributions of the superior parents is also very hard to determine. So the 2 schemes, well-known schemes like among HAPSIB selection and among and within HAPSIB selection were proposed, where we have a generation 0, where we have these initial parental population sizes that I mentioned. Okay, and then first cycle that where we have just basically a crossing block. So it's a computational crossing block, this generation 1. Okay, and this is all all these part bulk pedigree. These are all QGENE lingo, which we'll get into. And then because we have this 2 different strategies where you have one includes within family selection and the other one does not. Essentially, in this generation 1, in the case of among HAPSIB selection, we simply go into a trial, and then that trial goes back into the crossing block, and then so those same individuals get cloned, and then they go into generation 2. Superior parents are selected, and then the cycle starts again. Alternatively, or additionally, in parallel, we're conducting among and within HAPSIB selection, where we have the same selection pressure among family, but we add the within family selection pressure, and we'll look at what these various options are established. We can establish through our simulation. So Rejan has a question: in the case of study selection, is the heritability a broad sense heritability or narrow sense? So that's a very good question, and it's actually it is narrow sense heritability is the short answer. But in the case of simulations, we have to be a little bit more granular in the way we set that heritability. So I'll talk about that in a second. So here it is. So essentially, we have an individual line-based heritability as established here. But for QGENE requires per plot phenotypic values. So that line-based heritability needs to be broken down into individual as the average over n number of plants by years, by locations, by replications. And so these are the general formulas used, okay? But we will look at how, during the hands-on part, we will look at how these are actually applied. But essentially, that's what we want to drill down to. Once you know the narrow sense individual heritability, line-based heritability, we can then take those various components, and these can be obtained from anywhere from the literature or from your own data. And then from there we can calculate it so that we end up with a per plot error variance, which results in the heritability that we wish to simulate in the different environments that we are looking at. I hope that makes sense. So we can. These are some of the results, for example, where we have a series of combinations, okay, between parents and heritability, levels of heritability, and then the two different breeding strategies, okay, across 3 different genetic models, okay. So we have full additive, additive dominant, and full dominant, and the lines, the vertical lines here are the 2 breeding strategies, but they what they indicate, as you see, they shift depending on the scenario. What they indicate is the point at which 90% of the attainable genetic gain is accumulated. Okay, so if we take all of the attainable rate gain between cycles and add it up, and then extract the 90% and then determine at which cycle that is occurring, that's what those lines mean. Okay, so as you can see, depending on the strategy, on the genetic makeup of that particular breeding program, you reach that plateau a lot earlier in some cases, or a lot later in others. Okay, and or a lot later in others. For example, if your population, the impact of your initial population size has a large effect on when your breeding program plateaus. And this is an important piece of information, okay, for essentially establishing the long-term durability of the breeding program, or the sustainability of the breeding program under the assumptions of a closed system. And this is just a summary slide of that same information, but it just gives us the average rate of genetic gain per cycle at up to 90%. Once it plateaus, there's really up after that, since there is no gain, we want to understand what is the average rate of genetic gain up till the breeding program is essentially depleted of genetic variance or genetic gain. And so what this figure reports is just the average per cycle under all the various scenarios. So this is also a piece of information that in this case, in this particular analysis, we were also interested in determining, because the industry has goals in terms of pushing the rate of genetic gain, and we want to find out what are the different scenarios that enable us to reach that particular goal. And as you can see, although some of them might have that rate of genetic gain early on, let's say that rate of genetic gain is 2.5. Okay, some of those might allow you to reach that initially. On average, you might run out of a specific component in the genetic gain equation that might put the breeder below the expectation. So in summary, I have here, an equation and then two images. So one of them is the Goodyear blimp, and then the other one is the Hindenburg. What I'm trying to show is that the return on investment or return on effort is in my own terms, is sort of proportional to the risk that we undertake, which it has some proportion to the amount of successes, and minus the risk that we associated with the amount of failure, and then some projected gain. And that can tell us some information about what is the what is the potential return on the breeding program in terms of the genetic gain. And so that is the difference between reaching the upper ratio, right? Because that's what we want in this return on investment. What we want to is establish what is the average gain, and or what are the number of scenarios that give us on average an advantage, and what are the number of scenarios that on average give us a disadvantage, and then we associate these, we create a relationship between these two, and then we try to reach the upper ratio of these of the possible scenarios that we can simulate. And so therefore that this is where QGENE becomes useful. So it allows us to pose questions and remove some of the intricacy that is involved with plant breeding programs without having to obviously undertake the costs that would be potentially involved in empirically establishing this information. Okay. And then in the case of, for example, a new tool such as genomic selection, this can be logistically challenging. So we can use simulation to understand what the genetic gain is across this the implementation of these new tools. But it is not a tactical tool, so it just allows us to strategize and then make major shifts in the direction or guidance of the breeding program. Acknowledgements to my group here at Miguel, Seamus, our staff, both here and in New Zealand, as well as my grad students working in both simulation, but also a number of other areas, and undergraduate students as well, funding agencies that help with our work. If you have any questions, feel free to email me at the email here, or Jenny, which she'll be introducing her work shortly. So at this point, I'd like to switch gears and introduce Jennifer Lynn. She is a student in my group working on subjects involved with complex inheritance and complex traits in pulses. And one of her projects involves the utilization of QGENE. So Jenny, it's go ahead.
All right, so thank you everyone for joining us today, and thank you for the wonderful introduction of Valerio. So let me just share my screen, and hopefully everyone can see now.
Yep, all good.
Perfect. All right, so on to the fun part. I get to show you guys how to actually simulate a breeding program. So before I begin, I do want to mention that you guys can feel free to interrupt me at any time. We want to make this hands-on and with computer simulations and coding. There may be challenges, so at any time, feel free to interrupt me or message the chat. So let's begin. So this is the outline of what I'll be discussing with you guys today. We'll talk a little bit about QGENE. I'll give you guys a brief introduction. We'll talk a little bit about why we use it, and we'll go into how it works with the workflow. And we'll be going over all the files that we need to have in order to run a simulation. And we'll be doing simulations first with the engine and then with the module. And then I'll go over how we can actually process the simulation output files. And we can also look at some other output files that you guys may be interested in if you want to use QGENE for your own purposes. So as I'm sure all of you are aware, breeding programs can take a very long time to complete, can take up to a decade. On the right here, I have an example of the pedigree method. And as you can see, it can take between 8 to 10 years until a variety can be released. And at each stage, we can see that there are various actions being taken. And so with each of these steps, every decision that is made can greatly impact the success of the breeding program. So this is where computer simulations sort of come in. They can provide us with information that we would not be able to obtain empirically in just a matter of days. And there are a couple of simulation tools that are available, including GeneFlow, Gregor, and of course, QGENE. So QGENE is a stochastic simulation platform. And you might be wondering what exactly is stochastic. So in simulation theory, there are two types of simulation, either deterministic or stochastic. So with deterministic simulations, essentially the model does not allow for randomness. So with your inputs, each time you input something, it will generate the exact same output each time. Now, the opposite is true for stochastic simulations, which does allow for a level of randomness in the model, such that you can, each time you input your data, you can actually generate different outputs each time. And so this is not only useful from a statistical standpoint, but as well, it can better reflect what we see in reality. So QGENE uses something known as the ENK model, where E is the number of environments, N is the number of genes, and K is the epistatic network. And this allows us to simulate genotype by environment interactions, as well as epistasis. So with this in mind, let's talk a little bit about how QGENE works. So you start off with a QG file, and this contains information about what you want to simulate. So these are going to be all your parameters. Once you have generated that file, that gets run through the QGENE engine, which then creates a GS file or gene environment system file, as well as a population file, POP file. That, along with a QMP file, which specifies the breeding strategies, will be put through the QGENE Plus module, and that will produce a number of output files for you to analyze. So starting off with the QG file, this is where essentially you put all the information about what you want to simulate. So we have traits, and for the example we'll be doing today, we're going to be looking at seed yield. For the environments, we will be looking at 3 different environments just to show as an example: nursery, winter nursery, and field. For the error variances, this is to be calculated, but I will be showing you exactly how to calculate that. For the linkage map, we'll be using the consensus common bean linkage map reported by Galliano et al. From 2011, as we work with common beans. And for the QTLs of the genes, I have a number of seed yield QTL that we will be inputting into the simulation. The markers will come from the linkage map, and the populations will be generated by QGENE. Lastly, for diagnostics, we can essentially just indicate if we want a simple or detailed diagnosis. So let's now move to the RStudio Cloud. So if everyone can just access this website, and if there are any issues, please let me know in the chat, or just speak up and we can wait for you. So please go there now. So okay. So I will wait for about a minute or two just to make sure everyone is able to access the project page. And actually, I'm wondering if we can get a show of hands to see if people are able to access. Okay, yep, I'm seeing one hand here. Oh, okay, perfect. Okay, awesome. So it's looking like a lot of us are able to access. Yeah. So I'll wait a little bit longer so that we can get some more show of hands.
Are they just raising their hands?
Yeah, I'm just checking to see if everyone's able to access it. Okay. Okay. So I think that, yeah, I've seen some show of hands. So now let's actually get into the workshop. So once you've accessed the project page, you guys can go to copy of workshop files. And from there, you guys can actually open the example Excel file. So in the example Excel file, I'm going to actually go over the different sheets in that file and how we can actually change it to fit our own experiments. So the first sheet you'll see is the diagnostic sheet. This you can leave as is. It basically just tells you if everything has been input correctly. The next sheet is the environment sheet. The first column is the environment number, and this is fairly arbitrary. Oh, somebody has a question. Okay, so someone cannot access the file. That's okay. You don't need to access it. This is just if you want to follow along with me. If not, you won't really need to open it. Just keep listening and because I have the screenshots. So yeah. Okay. Yeah, so let me continue. So the second column is basically the name of the environment. And here we just have three environments, nursery, winter nursery, and field. And the frequency is basically how often it appears in the final population, and the frequency should add up to 1. The next sheet is the trait sheet, and this is where we can specify what traits we want to simulate. So the first column is the trait number, again, very arbitrary. You basically specify this so that QGENE can distinguish between which traits you're going to be selecting on. Next is the trait name, and so SY stands for seed yield, which is what we're going to be simulating. There's error within, error between, and error mixed, and we can just leave that as is since we're going to be looking at error in terms of heritability. So here is the error sheet, and I will be explaining how we get these values in the error within. Starting with column 1 is just the trait name. We also have the environment name. If all environments are the same, you can essentially specify 0. And of course, you have the error within, and like I said, this is determined from the Calc H2 sheet, which I'll be going into detail in a moment. The next sheet is the map sheet, and this is where we basically input our linkage map. The first column is where we can put the chromosome that the marker or QTL is located on. We also have the name of the marker QTL, the position in centimorgans, and the number of alleles. And the type is just to indicate whether you have a marker or QTL, so 0 for marker and 1 for QTL. The next sheet is the locus effects sheet, and this is where we input specific information about the QTLs. And this was obtained from the literature. So the first is the name of the QTL, the trait, the environment. In this case, we're going to specify 0 so that it's present in all environments. In some cases, you might have QTLs that are only present in one environment and not the other. And so this allows you to specify which one it is present in. The gene effect type, so 1 for additive, 2 for epistatic effects. To keep things simple, we're just going to have additive effects. The effect type, so you can actually specify values. And this is what we're doing here. So we've chosen 0 for effect type, and these are the effect sizes of the QTLs, again, obtained from the literature. The marker effect sheet will be all the markers that were found on your linkage map, and make sure that the names match up. Another thing to just briefly note for troubleshooting purposes is that locus names should be less than 8 characters if possible. You specify the marker type, dominant or codominant, and this you just leave as 0 for values 1. Next is the population sheet, where we can define either parental population or an error population. So we have the name of the file, we have the name of the population, and we have the population sizes. For the error population, anything over a size of 200 is adequate. In this case, we've decided to use a size of 500. Next, we have the population type. So 1 is to specify that it will be generated by QGENE. If you use 2 or 3, this is you can actually input your own populations. And lastly is just error use. So 0 for parents and then 1 for error. Because we've specified type 1 population, meaning QGENE will generate the population for us, all we need to do is just indicate the allele frequencies at the beginning. The remaining columns you can leave as is unless you want to change the order of alleles or sampling methods, but that's up to your discretion. So let's talk about the calculation of variances, error variances. So I'm sure you guys remember this slide from before. So I've summarized it on this slide here, and this is where we can actually calculate our per plot heritability based on our neuroscience heritability values obtained from the literature. Just so you guys kind of understand how the calculations are done, the phenotypic variance is based on 1 divided by the per plot heritability. The error variance is calculated by taking the phenotypic variance and subtracting the genotypic variance. And you end up with a per plot heritability by taking the genotypic variance and dividing it by the genotypic variance plus the error variance over the year multiplied by the number of plants per plot. And so all of this is in the Calc H2 sheet, and all you really need to do is just change some of the values, and you can see them there. So let's now go into our Studio Cloud and actually generate our QUGENE files. So all you need to do is go to the file generate QUGENE.r and copy everything from the script and paste it into the console. So let's do that. So generate QUGENE. I'm gonna Control A, copy, and paste. So once you've pasted it, you should see a number of files appear. And at the very bottom, you will see your QG file, and it will be titled example.qg. So going back here, the next step is to actually download some of these files, and one of them will be the QGENE engine file, which you will need to run the simulation. So with QGENE, it will be a Windows file. So if you are running on a Mac, unfortunately, you will not be able to run QGENE today. But I am able to run it on Mac. If you do have a Mac, email me after this webinar. So we basically want to select 5 files to download. We have the example QG file, my strategy QMP, QGENE, the engine file, QLine Plus, and the MIO file. So let's do that now. All you need to do is go to More, Export, and Download. So I'm going to select my strategy QMP. I need QGENE, QLine Plus, QLine Plus MIO, and lastly, example QG. And basically, I'm going to go to More. I'm going to go to Export. And all I need to do is just download that. So once you've downloaded the file, you can open that up. And you can actually go to Extract All and extract. Okay. So at this stage, I just want to kind of get a show of hands to see if everyone has followed along. Please raise your hand if you're able to, just so I have an idea. Okay, I'm seeing some hands, so that's great. Awesome. Okay. And again, feel free to just message at any point if you are having any difficulties, and we can wait for you. Okay. So, I am seeing in the chat, Thomas is unable to access the file. Thomas, are you still having issues accessing the files? Are you on the correct project page? Make sure you go to the link at the top. Yeah, so yeah, link has been sent again, and yeah, make sure it's the correct project page on the URL. If you're on the wrong project page, you will not be able to access the files. Okay, so I'm going to continue. Once you have extracted the files, you can now run QGENE. So all you need to do is double click QGENE and allow it to run. It'll say Windows is protecting your PC, but it's okay. QGENE won't have any viruses. You can run anyways. And so you should be able to run it. Okay. So from there, all we need to do is go to run batch job. Oh, okay. Let's close in a second. Yeah, that sometimes happens. Okay, try that again. Okay, can you see?
Yeah, but you have to share the whole screen.
Oh, okay. Yeah, okay. I'm gonna share the whole screen. Okay. All right. We're able to see everything? Perfect. All right, so within the engine, you go to Browse. We're going to select the example QG file. We're going to say Open, and all you have to do is press Run. And so from there, it should now be running the engine. And once the engine has finished running, it should say completed successfully.
So what is it doing?
Yeah, so right now it's taking all of the information in the Excel file, and it's basically, the QG file, and it's generating a gene environment system file as well as the population file. So now it has completed successfully, so we can close that. And so let me just get another show of hands to see if everyone was able to successfully run QGENE. Awesome. I see Paul has raised his hand. Okay. Thank you. Yep. Is there anybody who is having any difficulties? Oh, process. So someone is asking if I can repeat the process of running again. Yes, no problem. So yeah, I can definitely run that again. So what I'm going to do is I'm going to just delete the files. Oh, not that one. This one, this one, this one, this one. Okay. Yeah, so I can definitely run that again. All you need to do is, yeah, okay. So everyone can see, you can see my screen, right? Okay, perfect. Okay. So within the QGENE engine, go to Run Batch Job. Okay. Under Run Batch Job, you go to Browse. From there, you basically select your QG file, and all you need to do is press Open. Once you've done that, you should see the QG file appear in the engine list here. And once you've done that, you can just press Run. And so that will run it again. Okay, so I'm seeing some, oh yeah, so if it's taking a little while, that's okay. That's no problem. It might take a little bit of time. Oh, it's telling me there's an error. Okay, hold on a second. I think I have too many things going on at once. So that's okay. So if you are having trouble running the QG file, that's okay. Go back to the project page and go to additional files. And I basically have a QG file here as well as some of the output files. Okay, so let me see. Oh, I didn't include the GS files. Okay, that's okay. Okay, so I'm going to try running it one more time. I think I just have a lot of tabs happening at once. So give me a second. Okay, so I'm just going to run it one more time. Oh, okay, let's extract all, extract. Okay. Yeah, let me put a different one. Yep. Okay, I'm gonna re-extract the files. Give me a second. All right. So back to copy. So I'm gonna select my QG, QMP, QG, QN, MIO. I'm gonna go to more, export, download. Okay, open up the zip file. I'm gonna select extract all. Yes. And from here, we're going to run it one more time. More info. And batch job. Browse. We're going to select our example. And we're going to press open. And we're going to run it. And hopefully it will run smoothly. Sometimes when you have a lot of processes happening at the same time, you may run into issues. Okay, and this time it looks like it's running smoothly. Nope, it does not want to run. Okay. I'm not sure why it's doing that. That's okay. I have previous runs from before, so we're just going to continue with those files. Okay, so if it ran appropriately, we should see a number of output files, and I'm going to go back to the PowerPoint. I don't think this one. Sorry. I'm having trouble moving this thing for some reason. So I'm going to reshare because it's for some reason. Okay, stop share. Okay. Yeah, sometimes it happens. I think it's just because like if you have too many things running at the same time, it becomes problematic. Okay. So oh, there's a question. My PC is not allowing to run QGENE. Yeah, so sometimes you just have to go to more info, and under there, you can go to run anyway. If not, it's possible that you might have an antivirus software installed, so you might need to disable it. Okay. Yeah, someone's a little bit confused. Yeah, so again, this is all going to be recorded, so you guys can go back to it. And yeah, I think it's, yeah, sometimes if you have a slow computer, you may run into issues. Okay, so someone's asking to repeat steps. For the interest of time, I'm going to just continue. And I think, yeah. So I think we can just, yeah, people can watch the recording. All right. So once the files have been generated from QGENE, you should see an error pop file, the example diagnosis file, GS file, and the parental population file. So we have now obtained our GS and POP files, but we still need the QMP file. And just as a side note, again, you have an error population and a parental population. Make sure that QLine Plus is using the parents POP file. So let's talk a little bit about the QMP file. This file essentially contains the information on the breeding strategy that you want to simulate. And so, for example, we're just going to be simulating a simple mass selection scheme. On the right here, I have a figure that shows how mass selection works. Basically, you start off with the population and you select desirable plants, or you can remove any undesirable plants. And this can be done for a number of generations until you get to replicated trials, where you then release your best performer. So if you guys want, you guys can open the mystrategy.qmp file using Notepad or TextEdit. However, I do again have screenshots for everything, so you don't need to necessarily open it if you're having difficulty with that. And we'll be talking a little bit about how exactly we read the mystrategy.qmp file and how we can actually change it to accommodate our own experiments. So in the QMP file, you should see something that looks like this. And starting with the very top row, we're going to go over all the different arguments. So the first argument is the number of strategies you want to simulate. Because we only have mass selection, we're just going to put 1. For the number of runs, these are also similar to replications, and each run is essentially 1 cycle, and 1 cycle is essentially from the start from crossing all the way until variety release. For our simulation, we're just going to be doing 10 runs. So next we have the number of cycles we want to simulate. And so we'll be simulating 5 cycles. We also have the number of crosses we want to work with. In this case, we're going to be doing 100 crosses. And for the actual crossing block, we can actually indicate if we have our own crossing block design, a POX file. But to keep things simple, we will just be putting random crosses. So next we have the actual strategies and the parameters for the strategy. So the very top row, you can see strategy number, and again, arbitrary. You can basically allows QGENE to distinguish between strategies, strategy name, and the number of generations. The number of generations, just so you know, is not inclusive of the crossing block, or CB. And so from F1 to 8, because we have 8 generations, that's what we will have in our QMP file. So the first argument F1, that is our generation title. Oh, the light went out there. We also have the single cross. That's basically our seed propagation method. And there's a number of options you can choose from that QGENE allows for. There is super bulk, basically the method of harvest. We have the number of replications, and not to be confused with the number of runs. This one is the number of replications for your plots. Your plot size, in this case we have 50. The number of locations and the specification for the location. So for example, 1 indicates nursery. If we had field, it would be 3. So the next 2 rows allow us to indicate whether or not we want to perform selection. So the first row allows for among family selection, and the second row allows for within family selection. Notice how we don't do selection until the F5 generation, where we have the first argument being the number of traits to be selected, the trait number, in our example, trait 1 is yield, the selection model. For us, we're going to be using the top proportion, and there are a number of options again available in QGENE. You can select bottom, middle, even random. And you can also specify numbers or values. For us, we will just be specifying a proportion. So that's going to be our selection intensity of 20%. So after we have made our adjustments in the QMP file, we also have the MIO file. This basically just tells QLine Plus what files it needs to call in, and it needs to be in this order. You can modify it based on if you have different names for your files. So next, we can actually run the QLine Plus module. So at this stage, I'm going to go back to just sharing my screen. And okay. Okay. So from here, I'm just going to quickly delete the output files. Okay. So assuming everything went smoothly, you should see the example diagnosis file, the example GES file, as well as your parental POP file and the error POP file. So with these 4 files, you can then go into QLine Plus, and all you need to do is double click that, and it should be able to run everything. And so while it runs, I'm going to go back to actually the RStudio Cloud. And I'll go over again the steps that were taken. So I'm going to deselect everything. So in the beginning, what we did was we went to the generate QG.R, right? And all we needed to do was essentially copy everything and paste it into the console and hit enter. And once you have hit enter, you should not see any errors, and you should see the example QUGENE file being generated. So yes. Yes, yes, of course. So let me go back to that slide.
Did you see the question?
I did, yes, yes. Yeah, so basically, that is essentially your method of harvest. So when we say super bulk, that essentially is what we do in mass selection, where we take all of the plants and we harvest them all in bulk. So we are not separating them by families, we are bulking them all together and we're harvesting them. With bulk, we actually do distinguish between families, but within that family, we're going to be harvesting all of those, all the seeds at once per family. For pedigree, this is where we can actually specify whether we are choosing within families or even between families. So it allows for a little bit more specifications. And then with part bulk, we are only partially bulking our harvest. So essentially, it's just how are we going to be harvesting our seeds? Hopefully that answers your question. Yes, so which folder can we see all the output files? So this will be after running QGENE. So I'm gonna do the QGENE simulation, the QGENE engine simulation one more time. So let's do that because I think a lot of people are having difficulty with that. So, oh, another question. Are they separate softwares? So how it works is QGENE is the engine, and technically, yes, technically they are separate softwares. So with QGENE, it's very broad. So you'll need QGENE for everything you run. QLine Plus, it is a module, so you can think of it as sort of an addition. And there are a number of modules that are available. There's QLine, I think there's QMARS for marker-assisted selection. There's a couple more, QHybrid. And so these are the other modules that you can actually choose from. And each one has specific purposes. So you can see them as sort of additions, but the QGENE engine, that's sort of the baseline. Okay. Awesome. Okay, so at this point, I'm going to go back to the RStudio Cloud. So once we have generated our QG file, what we can do is we can download them. So we're going to select the QG file. We're going to select the QMP file. We're going to select the QGENE, QGENE EXE, QLine Plus, and QLine Plus MIO. So I'm going to wait until everybody has selected these files. Please raise your hand if you are able to do so. Perfect. I'm seeing some hands. Oh. Is anybody having difficulties or anything? We're just selecting files. Okay, so then we're going to go to more and export. So is everybody able to follow along? Please just keep giving show of hands or comment so that I know you guys are able to follow along. Okay, I'm seeing some hands. Perfect. Okay. So now we're going to go to download. Okay, so that will download a zip file. And you can either go to your downloads folder or click the downloaded file directly, which is what I'm going to be doing. Okay. So is everybody able to follow along? Please keep giving show of hands so I know people are following along. Perfect. I'm seeing hands. Okay. So from there, you're going to go to extract all. And we're going to say extract. So that will create a new extracted file. Oh, am I? Oh, my bad. Okay, sorry. Okay, so if you're able to extract the files, you should see something like this. Okay, I'm going to re-extract the files just so that everyone can see. So you basically go to your zip file and you go to extract all and say extract. I'm just going to replace them. Okay. So is everybody able to extract the files? Is anybody having any difficulties with doing so? Please comment. Okay.
So what do we have at this point? We have QG, we should only have these files, right?
Yes, yes. So you only have the five files. Basically, the example QG file, which we generated using the R script, and the remaining 4 files are already in the folder. So we're basically just downloading the files that we need to run QGENE. Okay. Yeah.
So at this point, we have the QG file, which is our G environment or genotype environment, and then we have our QMP file, which is our simulation our breeding strategy. Okay, so these are the 2 key files from which QLAN Plus will then conduct the actual simulation, generate the simulated population. So go ahead, Jenny. Sorry.
Yeah, no worries. Okay, so we're gonna run the engine one more time, and I'm gonna try to close a couple of things. I think there's just a lot of files open right now at once. Okay, so hopefully it'll run. I think sometimes it just runs into issues if there's too many things open at once. Okay, so we're gonna go to run anyway. Okay, and you can see my full screen, right? Okay, perfect. So once you double clicked the QGENE engine, we're going to go to run batch job. Okay. And we're going to select browse. From there, we can choose our example QG file that we generated. We're going to press open. And then we're going to run it.
Run this again.
Yeah, because I think people were having issues before following it. They wanted me to go over the steps one more time. Yeah, so. Okay. So are people able to run the QGENE engine? Do we have any issues with running it? Is anybody having difficulties? Show of hands if you're able to run the engine. Okay, perfect. I'm seeing some. Awesome. Okay. Okay. So I think at this point we'll move on, and if people still have issues, they can go and view the recording. Okay. So just gonna skip through here. Okay, so once you've run the QGENE Plus module, you're gonna see a number of output files, and they'll all start with Z out. So from there, we can upload each of these files individually. And I'm just going to show you how to upload it. But in the additional files folder, these files are already available to you. So all you would need to do is just copy paste those files into the copy of workshop files. And so we're only going to be focusing on 4 files in terms of output processing. These are the fit file, fix file, ham file, and var file. So let's talk a little bit about what each of these files does and what they can be used for. Before I do that, I will show you guys how to quickly upload these files. So back to our Studio Cloud. Within our, I'm just going to go and select this. So,
So they ran our QGENE Plus now.
Yes, so once QGENE Plus has been run, they can now upload those simulation output files back into the cloud so that they can actually process it. So go to upload, select choose file, and we're going to go to the fit file here, and we're going to say open and okay. And so you'll need to upload these one by one because the cloud only allows you to upload zip files if you want multiple. So we did the fit file, we're now doing the fix file and say open. Okay. Do it again, so upload, choose. Now we want the ham file, so let's open. And one last one, choose. No, the bar file is not there. Maybe it will be in here. Not sure where my var file went, but that is okay. This will all be in the additional files page. You can just copy all of these, go to more, go to move, and select here, and you can choose. Okay. So, so back in here, we should now have our fit file, fix file, ham file, and var file. So at this point, show hands if everyone is able to upload or move those files into the copy of workshop file. So a quick show of hands, or if anybody's having difficulties uploading or copying files, please send it into the chat. Okay, I do see a hand. Please comment if you're having any difficulties. Okay. So once we have, oh, somebody raised their hand. Okay, that's fine. Okay. Before we continue, I'm going to talk a little bit more about the actual files and what their contents are. So starting with the fit file, this basically gives us the fitness or genotypic values reported for each cycle. And so starting in the first column, we have the model number. In this case, we only have one model. We have the strategy number. We have the run number, cycle number, the environment, the trait, and of course, the most important, the fitness or genotypic value, as well as the adjusted fitness or genotypic value. And I will talk about how that is calculated. So like I said, the fit file has the average fitness or genotypic values for the population, and it can be used to estimate genetic gain. So the adjusted fitness is calculated by taking the fitness and subtracting the lowest target genotypic value, and then dividing that by the highest target genotypic value minus the lowest target genotypic value. And then you multiply that by 100, and it essentially converts it into a percentage. This can then be used to calculate the adjusted genetic gain. And so basically it's the adjusted fit file for each cycle minus the adjusted adjusted fit value for the cycle prior that. So we also have the fix file, which contains the percentage of favorable and unfavorable alleles that are fixed in the population, as well as the ham file, which contains the Hamming distance for the population. The Hamming distance can be described as how much an individual differs in terms of marker alleles from the target genotype. And so a smaller Hamming distance means that an individual is closer to the target or ideal genotype. Basically, the smaller the Hamming distance, the better. We also have the VAR file, which contains estimates for the genetic variants. So within the VAR file, it outputs all the different values for each cycle and for each population. So the one we're going to be looking at specifically is for the field, and we're looking at the additive genetic variants. So it will be called var add. So from here, we can now go to the script called process output R. Just to keep in mind, go to the copy of workshop file. So do not go to the workshop files. It has to be copy of. And from there, all we need to do is, again, paste all the code from the process output into the console and then hit enter. So let's do that. So we're going to go to process output. We're going to say copy and paste, and then we're going to press enter. So once you've done that, you should see a PNG file. Oh, whoops, my bad. Okay, let me try it again. Okay. Okay. So let me do that one more time. You go to the process output.r. You're going to copy everything from the script. You're going to paste it into the console and hit enter. And once you do that, you should be able to see a PNG file pop up, and you can just click on it and you should see the processed data. I do want to mention a little bit about the code. I think it'll be interesting to kind of see, you know, for your own reference, what exactly goes into the code. So these are the different packages that need to be installed if you do want to run this code on your own computer locally, and you call it those libraries. So this is where you can choose your working directory. You input your trait of interest. For us, we have seed yields, and you can change that if you have different traits, if you have plant height or days to flowering. You can also input the environment. In our case, we're interested in the field, but if you are interested in other specific environments, for example, if you're interested only in rain-fed, or if you're only interested in irrigated, then you can change those. This code here basically just specifies what the file is, has not read in the file yet. It's just indicating what the file is. And this is where we actually read in the file here. This here is just to ensure that the columns are either factors or numeric. From there, we can actually determine the genetic gain. Essentially, I take one column, I basically shift it down by 1, and then I subtract one column from the next and to obtain the genetic gain values. And then we can summarize it based on means. We can obtain the standard deviation and also a standard error. And from there, it would produce a plot. And that's all the code that goes into creating the plot. And so the rest of the files is quite similar, where we have the, where we can actually produce the plots. In the end, we have here where I just arrange all the plots in one single organized plot so that you can see everything. And then this one will just save it as the PNG file that you see. So at this point in time, do we have a show of hands of people who are able to obtain the PNG file? Yes, okay, awesome. Okay. Yep, so that's awesome. And what you'll likely notice is that each one is going to be, well, actually in this case, because we are using the same files, unless of course you have managed to run QGENE and Q1+, then you'll see different outputs. So from there, you'll see something that looks like that. And yeah, so that concludes the tutorial. I now want to just go over some of examples that I've developed from my own research. So, oh, actually, before we get into that, just a couple of output files that people might be interested in. This is actually the POP file. And what we can see here is the actual markers. And it's also separated by each linkage group. And it will basically output each and every different marker. And so each of these rows is a single individual. We also have the POU file for those who are interested. This produces outputs. Oh, I think there's somebody in, oh, okay, that's just you, okay, sorry. This produces inputs for the genotypic value, phenotypic value, as well as the Hamming distance for each individual. Yeah, so now I'll talk a little bit about some of the examples that I have from my own research. So for my project, I was comparing 5 different breeding strategies, specifically bulk breeding, mass selection, single seed descent, pedigree method, and the modified pedigree method. And this figure here just shows the trend of genetic gain. In this case, we see that over time in a closed system, genetic gain decreases and eventually plateaus. The black line is the average for all these strategies, and we can see how the strategies sort of compare. If you look at the conventional breeding methods for days to flowering, DF, we can actually see how the pedigree method is achieving genetic gain in much fewer cycles than the strategy average. And we can see how that differs between different methods, either conventional, speed breeding, or genomic selection, and between traits. Oh, yes, I have questions. So does this program work for crops and traits that you do not have linkage information on, but you have relationships between markers and traits? So it's a very good question. I think at the end of the day, you will still need to have a linkage map in order to run QGENE. However, you can supply recombinant fractions rather than the positions or the in centimorgans. So yeah, I think you will still need to have a linkage map. Yeah. Do you have anything to add, Valerio, or?
Yeah, that's a good question. So the whole reason why QGENE requires a linkage map is because it needs to be able to establish, of course, recombination fractions in order to then simulate basically segregation and segregating populations with different recombination fractions that you established through the original linkage map. So you have a couple of avenues. I mean, if you have, say, physical distance information, then from there you could potentially create a linkage map, right? It depends on the type of population that you use, etc. But that would be one avenue. The other avenue would be that to use the actual physical information directly, but there's going to be some. I would say you're sort of overriding the assumption that those polymorphisms that are present there are somehow reflecting the recombination rates, which would not be quite accurate. So the best thing would be is to if you only have physical information, would be to try and then create a linkage map from there. Now your question says, does it work for crops and traits that you do not have linkage information, but you have the relationship? So by that, I assume you mean you have you only have, say, the QTL or the GWAS hits alone, and you don't have the linkage maps. Is that right, Paul? Right. Yeah. Well, in theory, if you only have the GWAS hits, no, that would mean that you're only inputting the sort of the QTL side of the information and not the neighbor. So you do have to have some sort of map on which you're placing the QTL, the effects. So those effects, whatever, if you're using QTL or genes, however you're presenting that information on, but it has to be placed somewhere on a genome, whether that is a linkage map or an actual genome. Hope that answers your question.
All right, so I will continue. All right, so yeah, let's move on to the next figure. So this figure is showing the proportion of cumulative genetic gain from the total for each cycle. And we can see at what point it essentially plateaus. So essentially by cycle 5 for conventional methods, we see that we've already achieved 96% of the total cumulative genetic gain. So with that in mind, we decided to look at how many cycles it would take to get to that 95% cumulative genetic gain. And so just to also keep in mind that this is averaged across all strategies. Oh, let me see. Oh, rippling effect. I'm not very familiar with the rippling effect, so you may maybe it's answer that.
So if I would the question on rippling effect, it's a good question, but QGENE does not. If I understand correctly, the rippling effect is the way that it's involved in the way you construct the linkage map, and so QGENE does not build a linkage map. It just assumes that that you've already gone through all the processes of of assembling the linkage map. So it doesn't really verify or test whether those distances are are affected by recombination fraction at all. So it doesn't affect it in that sense. It does not affect the simulation output. Does that make sense? Go ahead, Jen.
All right. Yeah, so from there, we wanted to look at how many cycles it would take to get to 95% of the cumulative genetic gain. So that's what this figure shows here. We have the number of cycles, and we can see how they vary between the strategies, between traits, as well as between the different methods, whether we're using conventional methods, speed breeding, or genomic selection. On the right, I also have the relative genetic gain achieved per strategy for each of these scenarios as well. And so we do see that there are differences, although differences are not pronounced in certain ones. And in genomic selection, we do see that these differences are more pronounced. So this here is the the fixation of favorable alleles. And so this will be similar to what we generated today, where we're showing how many alleles are getting fixed, and we're focusing only on the favorable alleles. And so we see that there are different rates of fixation between the strategies. This is showing a plot of the Hamming distance. And so similar to what we saw in the fixation of favorable alleles, we're also seeing different rates of change when it comes to the Hamming distance. So over time, we're seeing that the population is improving, the Hamming distance is decreasing, but how fast that Hamming distance decreases does vary between the different strategies. And this here shows the genetic variance after the first 2 cycles. And genetic variance is very important because it ensures that in our population there is still diversity. And so we can see that between the different strategies, they decrease at different rates. And something to note is that with strategies like single seed descent, the pedigree method and the modified pedigree method, in certain scenarios, they do decrease quite rapidly. And lastly, this is just showing principal component analyses to show the variation in the 5 different breeding strategies by looking at how they vary in terms of effective population size, the fixation of favorable alleles, Hamming distance, and of course genetic gain. And we do see various clusters beginning to emerge. There seems to be an interesting cluster forming regarding speed breeding and bulk breeding, as well as bulk breeding and genetic selection. We're seeing them sort of cluster separately. So this kind of gives us an overview of how the strategies sort of compare with each other. We're also seeing that the single seed descent method, pedigree method, and the modified pedigree method show some overlap. They don't cluster together, although the pedigree method sort of has certain clusters emerging as well. Yeah, so I would like to thank, of course, my supervisor, Dr. Valeria Oyes-Villegas, my committee member, as well as the lab members who have been very, very supportive. You guys can check out our website at www.pulsebreeding.ca. And I also like to thank collaborator Dr. Vivi Ariev from the University of Queensland, and of course, our funding partners. So thank you very much. And I'm happy to answer any questions you guys have.
Thank you, everybody, for your attendance and for joining us in this workshop. We have a little bit of time. If anybody has any questions about anything that we've covered.
Thank you.
I think I've posted the emails, our emails there in the chat. So if anybody has,
if anyone has questions, feel free to email me. And especially if you want to perform simulations with a Mac, please feel free to send me an email because that's a little bit different.
As one thing, this was obviously a very simplified run of QGENE. As you saw, there's many, depending on your particular in silico experiment, there's many fine details and variables that you might want to define and specify to fit your question a lot better. So this is just a sort of way of getting you started. But if you have any questions to basically get your question forward or get your experiment forward, don't hesitate to contact us, and maybe happy to help.
I think there's a question there about GWAS data and how it can be used in QGENE for clonally propagated crops. Yeah, so QGENE does have options where you can basically use cloning as the method of propagation. So I don't see why it can't be used for clonally propagated crops.
Yeah, in fact, that case study that I showed at the beginning, that was from perennial ryegrass, where a lot of the parents are actually cloned vegetatively. So it is possible to basically clone the genetic makeup of individuals throughout your strategy. Yeah, and GWAS, regarding GWAS data, as long as you have the effects, it's really all that matters, right? So if it becomes a bit more tricky of how do you place them onto a genome, because you do need to provide the software with some idea of how recombination is going to occur in these populations. So it needs to know from the linkage map establish what's going to be the potential for recombination and segregation in the population eventually. So the effects are one thing, and then you have the map on the other side. But definitely you can use GWAS data as long as you have the effects.
One last thing I did want to mention is, so some of these slides I have listed all the different parameters and what they mean. You can actually download that from the RStudio Cloud. So I'll just quickly show you how to download that.
Oh, there's a question on the Q&A as well. Yeah, so I can give you the file for the parameters. So if you go to copy of workshop file, go to the cheat sheet PowerPoint, and you can just download that. You can just go to export and then just download it. And if you open that up, it basically kind of summarizes all the different parameters that we covered here today. So all the different components of the Excel file, as well as the QMP file, which we are basically the ones that you're going to be specifying in order to make it so that it will work for your experiments. Yeah, as well, you can go to the QGENE manual as well, and they actually cover in greater detail than I have. Again, this is a very simple simulation example. So if you want to go into more advanced details, you can actually go to the QGENE manual, and I'm gonna see if I can send that into
the chat so that everyone can access it. So
Paul has a question on the Q&A. What if we can expand on speed reading versus conventional?
Oh yeah, so that was for my project, and yeah, because of time constraints, I didn't want to show you guys. Well, it would take a long time to actually simulate speed reading and conventional. So for speed reading, we essentially do post-analysis. So after running the simulation, we make adjustments to the actual cycle time. How speed reading works is basically it is a method that can increase the rate of development in crops through increasing the photo period. So there have been a few experiments done, I think it was in peas and I think in wheat, where they've actually been able to rapidly increase the rate of development so that it basically saves time. For our case where we're looking at common bean, dry bean breeding, it shortens the generation time from 8 cycles to 5 cycles. Sorry, it shortens it from 8 generations to 5 generations, not cycles. So that's speed breeding versus conventional. And okay, yeah. Hopefully that answers your question. And
is there a possibility of exceeding some values in a population better than the best genotypic responses with respect to Hamming distance? So if I understand the question correctly, is there a way to specify, to look at the genotypic response to the best Hamming distance and then set the parameter so that you exceed that genotypic response? If I understand that question correctly, yes, that would be possible. Yeah, you can essentially, all you have to do is just calculate, is just input your error so that based on that output information, you generate a new simulation exceeding those values, and you can definitely do that. Yeah. Jennifer, can QGENE be used to find genomic associations to traits?
like correlations?
Well, can it be used to map traits? As I understand the question. Can it find genomic associations?
So that I'm not actually too sure of, because I think when we do run the simulations, these are based off of traits or like QTLs that are known in the literature. Yeah, so I'm actually not sure. Maybe you can answer.
Well, there was that one case study, that one paper I showed at the beginning of the workshop, where they didn't necessarily map a trait, but they did simulate a set of situations, and then a set of populations, and then based on that those different scenarios, they would be informed as to what is the power of a particular experiment to detect a QTL of a particular size. So it might not, it won't tell you, it won't actually map traits, because, well, you're dealing with stochastic simulation based on a randomly generated distribution, but it can inform you as to what might be your chances of actually mapping and detecting an association. So it's basically, in terms of experimental design, it can inform you quite a bit. If going back to the speed versus conventional, and also including genomic selection, for example, in the work that Jennifer just showed, and which will be published here in the next few months. Yeah, if you have any further questions or interest, don't hesitate to contact me, and we'd be happy to guide you to get your simulation conducted. There's another one there.
If we find some low log quantity for complex traits, can we predict based on simulations the high log threshold possibility in future generations or in differently designed populations? So I think at the end of the day, well, with the high versus low log scores, I think that's more just so like your peaks and how likely that marker is going to be associated with a trait. So with a low log score, you can simulate it. I don't know if it will necessarily give you possibilities or in predicting future generations, but you can definitely input any low log value QTLs. Yeah, I don't know if I understood that question correctly. If you have anything to add, Lyra, about the prediction for future generations, I think.
So if I understand the question correctly, can we predict based on simulation high threshold or different? Yeah, I mean, with some post hoc analysis, you could probably determine, you could probably predict based on a particular log threshold. Yeah, if I understand correctly, your question is it basically possible to then say, what are the conditions or what are the scenarios where a different population would then result in a specific log threshold? You can definitely do that. Yeah, QGENE doesn't do it directly, but it will certainly give you the inputs to then conduct, say, the permutation analysis to determine what the log threshold would be. You can do that later on in ICA mapping or any of the other softwares. The presentation, the recording will be made available on YouTube and through our cloud, our Studio Cloud page. You have access to all of the files that you need to basically rerun the workshop.
Yeah, and you can download the cheat sheet file. Basically, just gives you all the different parameters so that you can change and what they mean.
And if you find any value in these types of workshops, don't hesitate to let us know. It's always interesting to know what you're interested in and what brings value to your work as people and working in plant breeding. And it would be interested to know what sort of other topics you'd be interested in touching and discussing. And just remember that these are all provided to you through the National Association of Plant Breeders. And so I encourage you that if you things like this are useful to you, to join an APB. And we'll give it a minute for questions. And if there are no more questions, we'll stop there and thank everybody for. It doesn't seem like we have any questions. So well, thank you everybody for your attendance, and we hope to hear from you and to see you soon at some point.
Thank you very much.